Hi, my name is Aina Jahabiti Aziz, student ID 2321912. So today we'll be talking about a research article. Title is Identification of Natural Antifungal Drug Candidate Against Ginger Soft Rat Disease. Computational Drug Design Approaches. So let's get to know the problem. So ginger is an important crop known for its nutritional, medicinal, and economic value. However, it faces a major problem, which is soft rat disease. Okay, soft rat disease causes the rhizome to rot, produce a bad smell, and turn the leaves yellow. So this disease is mainly caused by fungus called Phytum alphanidatum, which thrive in humid and moist environments, especially during transport and storage. As the infection progresses, it not only reduces the market quality, but also reduces its phytochemical and medicinal properties, including valuable bioactive compounds. So the key factor behind this problem is the secretion of NEP1 proteins by the fungus. So those proteins damage plant cell membrane, causing tissue death. So currently, farmers rely on chemical fungicide to avoid this problem. But excessive use of chemicals can lead to environmental pollution, health risks, and the emergence of resistant fungal strains. So that is why this study aims to find eco-friendly, natural antifungal alternative, particularly flavonoids and terpenoids from plants. So to do this, researchers use computational or in silico methods such as molecular docking, molecular dynamic simulation, and virtual screening to predict how these natural compounds interact with the fungal properties. So you will get to know further explanation on the next parts of this video. So thank you.
In this study, the protein targets related to the ginger soft root gene were obtained from the Protein Data Bank and prepared using Discovery Studio 4.0 by removing automolecules, ligands, and heteroatoms. The active sites of these proteins were identified using the receptor cavity method in Discovery Studio. The protein structure was then refined using the Galaxy Refine server and validated with Ramachandran plots and ProSA analysis to ensure accuracy. For visualization, PyMol software was used. A total of 55 bioactive compounds from Azadirachta indica were collected from the IMPPAT database and optimized through energy minimization using the universal force field. The protein's active pockets were predicted using the CASTP 3.0 server, and receptor grids were generated in PyRX. The molecular docking was carried out using Autodoc Vina, while Discovery Studio Visualizer helped analyze the binding interactions between the proteins and ligands. LTMA properties such as solubility, bioavailability, and absorption were evaluated using Swiss LTMA. Toxicity analysis was done through the ProTox2 server to predict safety profiles. Finally, molecular dynamic simulation using Desmond software and MMGBSA energy calculations were performed to study the stability and binding strength of the protein-ligand complex.
For the results of our study, we first validated the structure of the NLP protein before running the docking test. As shown in Figure 1, the Ramachandran plot shows that most amino acid residues are located in the favorite regions, meaning the protein model is stable and reliable for computational analysis. Next, molecular docking was performed on 55 phytochemicals. From these, 3 compounds showed the strongest binding to the NLP protein: quercetin with negative 7.7 kilocalorie per mole, negative fluorine with negative 7.6 kilocalorie per mole, and lutein with negative 7.4 kilocalorie per mole. These negative binding energies indicate favorable interactions. Figure 2 shows that these compounds form hydrogen bonds and hydrophobic interactions that help stabilize the binding within the active site. We then evaluated the biological properties of these compounds. As shown in Table 1, all 3 compounds displayed acceptable solubility and good predicted absorption, suggesting they could function effectively in biological systems. To test binding stability, we ran a 100 nanosecond molecular dynamics simulation. The RMSD plot in Figure 3 shows that the complexes reach a stable structure near the end of the simulation, which means the compounds did not detach from the protein. The RMSF plot in Figure 4 shows that any structural movement mostly occurred in flexible regions while the binding site remains stable. Finally, the interaction mapping in Figure 5 and the ligand torsion analysis in Figure 6 both confirm that the compounds maintain stable interactions throughout the simulation. Overall, these results suggest that Quercetin, nicotinamide, and rutin are strong natural antifungal candidates for inhibiting the NLP protein involved in ginger soft rot disease.
In the discussion, I will explain the key finding and interpretation from the computational analysis. The study focused on identifying natural antifungal compound from Azaricta indica to exhibit the necrosis including NLP protein and Pythium alpha-dermatum, the main pathogen causing ginger soft rot disease. The aim was to evaluate how well this natural compound interact with the target protein and assess their potential as eco-friendly antifungal agent through molecular docking ADME analysis and molecular dynamic simulation. Based on molecular docking result, 3 compounds were identified as the most promising inhibitor. First is quercetin, nicotinamide, and rutin with binding energy of -7.7, -7.6, and -7.4 kilojoules per mole respectively. Negative binding energy value indicates spontaneous and energetically favorable interaction between the compound and the NLP protein. The binding was mainly stabilized through hydrogen bonding along with hydrophobic and pi-pi stacking interaction. This combination have anchored the ligand with the protein site, suggesting that this compound can block the NLP necrotic activity and protect ginger tissue from fungal damage. The ADME analysis further supported the potential of this compound as natural drug candidate. The RMSD plot revealed only minor fluctuation, while RMSF values indicate that the active site residue were highly stable. This result suggests that quercetin, nicotinamide, and rutin are promising natural antifungal agents that can potentially inhibit Pythium alpha-dermatum that reduce ginger soft rot disease. The finding aligned with the global effort toward green chemistry and sustainable agriculture, showing that computational chemistry can be a powerful tool for eco-friendly drug discovery. However, it's important to note that these results are theoretical. Further research should include in vitro enzyme inhibition assays, in vivo antifungal testing, and structure-activity relationship study to confirm and improve the compound antifungal performance.
In conclusion, to address the overuse of chemical fungicides, researchers use advanced computational techniques under the framework of computer-aided design to identify potential antifungal agents. Through this comprehensive in silico analysis, there are 3 promising compounds such as nicotinamide, rutin, and quercetin that were identified as such promising inhibitors. These computational findings not only enhance the molecular understanding of the promising inhibitors, but also provide a foundation for developing environmentally safe and effective antifungal strategies to mitigate ginger soft rot disease. So that's all from us. Thank you.